All right, folks, I'll do a little chapter 3 run through here, hit some of the highlights, talk about some things that I think are important for you to know, especially for things like short answer questions and things like that. But anyway, chapter 3 is about the cell. Some important things to focus on, obviously, are these certainly the headings, right? So I mean, you're not going to talk about or give a heading like spontaneous generation without some things being important there. But really, the important thing about this is that early on, we used to think I would say scientists, but certainly the general population as well, used to think that organisms just sprang into existence, right? And so here they give you a little example of that. So if you had meat just left out, it would spoil. And so the idea was that life just sprang into existence on the meat for no other reason until they noticed that if they treated food in a certain way, maybe it didn't rot as quickly. And so they walk you through some of the processes that we took in order to reach the conclusions that life is not just springing spontaneously, and that it's actually coming from other life. And then this goes on to build what we call cell theory, right? And so you can read through some of the individuals here, but ultimately the kind of nail in the coffin for spontaneous generation ends up being Pasteur. And so you've probably heard that word before. So pasteurization, that's where this comes from. And so what Pasteur did that nobody else did before was found a way to allow a broth to interact with air, but in such a way that it couldn't deposit right directly in. And so he did that with these swan neck flasks. So he had a broth in the bottom of the flask. He would heat it up for a pretty long period of time. The steam was able to escape, and then when it cools back down, so air can go down into this part of the flask, but anything that's in there is going to be deposited. Anything that might be in the air would be deposited here, right? We wouldn't expect bacteria or viruses necessarily to be able to travel all the way through this tube back up to the top and down in. And so anything that comes in would settle here at the bottom. And so what he noticed is after he heated this broth up for a long period of time, left it here, he got no growth in the flask. Nothing at all. Everything that one would need for life was there, right? Nutrients was there. It wasn't just pure plain water. So if something were to be living, it should have been able to grow there. And so what he found is that wasn't the case. Then what he did was he would come back and break the top of the flask off and let it sit, and then he would get growth. The other thing he would do is he would take the flask and tip it over after a certain period of time, collect some of this material, tip it back, and then he would also get growth. And so that told us, it showed us that something in the air is what's causing the growth, and it's not just life springing eternal from whatever is in the broth, right? Whatever organic products are there. And so that was Pasteur's experiments. He did a number of things that contributed to microbiology, but this was certainly one of the kind of groundbreaking, or it was certainly one of the nails in the coffin for spontaneous generation. Right, and then that gets us to modern cell theory. When you read about cell theory, so all cells come from other cells, and cells are the fundamental units of organisms. Sounds like common knowledge to us now, right? But we have to remember there was a time when that certainly wasn't common knowledge. Honestly, before Leeuwenhoek, we had no idea that level of organization, that cells even existed. And when he first saw them, Leeuwenhoek, he called them animalcules. Had no idea what they were, right? And so actually the term cell, we saw cork first, and it looked like little prison cells, and so that's where the name kind of gets, or the origin of the name, right? And so it took us a while to figure out what cells were. And here you can see a lot of these time periods, 1800s. Again, in the grand scheme of things, it's just not that long ago. And that always just kind of catches me as interesting. So we figure out that what cells are. We figure out cells come from pre-existing cells. Anything living has to be made up of a cell. So when I talk about the fundamental unit of a living organism, that is a cell, right? Some students would, if I ask that question, what's the smallest thing that can still be considered living? Some students will put atom, I think, because they hear me say, what's the smallest thing? Remember, atoms aren't considered living, cells are. And so the cell is the fundamental unit of a living organism. Also keep in mind that viruses aren't cellular. Right? It's another reason that we don't consider them to be living. And then, so we continue on and we study the properties of cells, and we come up with this endosymbiotic theory. And I think this is put a little ahead of where it needs to be in your book, because the idea of the endosymbiotic theory is that prokaryotic cells, so cells that have no nucleus or membrane-bound organelles, they were actually engulfed by other cells and then used. And so one of the examples of that would be, and they're showing these as green here, so an example of that would be a chloroplast. So chloroplasts, if you look at them, if you don't know what a chloroplast is, that's the organelle that captures light in plants, and then is able to convert that light energy into chemical energy and make sugars, for instance. But if you look at that organelle in and of itself, it looks very much like a prokaryotic organism. It looks very much like a bacterium. It's got circular DNA like bacteria do. The cell membranes look very much like bacterial cell membranes. And just the more and more we study that, it looks like it used to be its own microorganism, and that it was engulfed by another cell, and instead of broken down and used as food, it was held on to and used to make energy for a cell. Same thing is true for the mitochondria. Right? Mitochondria is able to take sugars and convert that into ATP. If you look at a mitochondria, membranes look very similar to prokaryotic membranes. It's got circular DNA just like prokaryotes do. It divides through fission just like bacteria do. And so it looks like it used to be its own free-living organism that was engulfed. And so that's the endosymbiotic theory. Right? So this idea that one organism engulfed another, and that kind of developed into the chloroplasts or the mitochondria that we know today. The first idea, when Margulis kind of presented this idea, she was kind of laughed at, and there's no way that's true. But more and more, we're finding out that everything just lines up. So her hypothesis was those things were engulfed, and so let's test that. DNA, cell membranes, the way it divides, just on and on and on, the ribosomes, everything just kind of lines up that indeed is what happened. And then we can talk about germ theory, right? So again, this seems so obvious to us, right? You wash your hands, before you eat or before you do surgery, or you glove up in sterile environments and all that. Seems super obvious to us. But there was a time when that stuff was not obvious. And thousands, millions of people probably lost their lives just because physicians didn't know to wash their hands before they performed an autopsy on a cadaver and then come in and deliver a child in childbirth, and that child dies. And so that was one of the, so Semmelweis, there's actually a podcast that talks about him that's super interesting. I'll see if I can find it. But one of the things he noticed is that when physicians would deliver babies, the death rate of those were higher in the infant and in the mother than in other facilities, right, where maybe midwives or something were delivering. And so what he observed is that what I just mentioned, so the doctor would come out of an autopsy, wouldn't wash their hands, didn't know to, and then would deliver a baby. Maybe the mother gets an infection and dies. Maybe the infant gets an infection and dies. And so again, this is another one of those where he proposed, probably should be washing your hands before you interact with another individual. People were like, this guy, what's he know? But then more studies confirm that that's probably a good idea, right? You should probably wash up. You should probably clean the instruments between patients, right? And now that's just common knowledge. But again, not so long ago there's his lifespan, 1818 to 1865, and so it's an interesting bit of history. Right? And so you can read kind of how we process through things like with Jon Snow, not that Jon Snow if you've watched Game of Thrones, but how Jon Snow was one of the first to do an epidemiological study. So they were looking at, so it was cholera in London, and what he did was look at a map and see where all the cases of cholera were, and he found that there was a higher number of cases around one particular well. They shut that well off, and the cholera cases went away. And so that's also an example of kind of early observational science, and why observational science is not, some people kind of think you shouldn't be doing observational science anymore, but if you think about things like, so one type of observational science would be you go out into, maybe the jungle and you find new plants. But people will counter that with experimental science, right? Well, without the observation, you can't do the experiments. And so observational science is still pretty important, right? But now we know, so we have all these disinfectants and antiseptics, and we're kind of on the other side of this now, right? So we've used, so if you think about antibiotics too, but even antiseptics and disinfectants, we have to alternate using those now, right? Because things will build up immunity to certain types of, and so we have to be careful and make sure we include rotation into plans for using things like this. But yeah, super interesting science there. And then Robert Koch, he came up with the Koch postulate. So again, this idea that certain things cause certain illnesses seems obvious to us now, right? And I mean, most people, it's so obvious to us, you don't even get it sometimes, right? So I've had students come to me and say they had strep, right? And then later in the semester, we would talk about Streptococcus bacteria, and you could almost see the light bulb go off. Oh, they said I had strep. I didn't realize that term strep came from the Streptococcus bacteria that's causing the problem, right? Causing strep throat. And the reason we know that is through these Koch postulates. So the Koch postulates are essentially kind of a list of things or steps that we would go through in order to find out that this is indeed the organism that's causing the illness. And essentially what you would do is you would have someone or something that's exhibiting some symptoms. You would try to take a culture, right? So if I think that Streptococcus bacteria is causing strep throat, then obviously I should be able to culture strep bacteria from someone's throat. I should be able to then take that bacterium, and if I was to infect someone else with it, they would exhibit the same type of symptoms, and I would be able to, again, swab them and remove strep bacteria. That was kind of Koch's postulates. Now, obviously, you can see there's some problems with those. For instance, I mean, you're not going to take strep from somebody and give it to somebody else just to see if it really is strep. You might could do that in mice, right? But you're not going to do that in humans. So there's some problems with Koch's postulates, but what it does is it gives you kind of an entry point to ask some questions about possible pathogens, and then develop some experiments in order to see if it is in fact that organism that's causing that problem, right? But that's Koch's postulates, and you can read through those as well. Here's another example, and for those of you that are going into maybe radiography, or if you're going to be looking at x-rays, this is a common one now, right, with COVID. So seeing either clear lungs or lungs with pneumonia, right? And so pretty much should see black in the lungs, so they should be filled with air. And so when you start to see them as being opaque, that's the problem, right? So a lot of infection here. This one is specifically pneumonia, but all sorts of things why you would see opaqueness in the lungs in an X-ray. Right? And then your book goes on to talk about some of the differences between prokaryotes and eukaryotes. Major difference would be nucleus, right? So prokaryotes don't have a nucleus; they just have this nucleoid region. So if you think about that term nucleoid, what is a humanoid, right? It's something that's human-like but not really human. So a nucleus or a nucleoid is something that's nucleus-like but not really a nucleus, right? So there's no membrane here. So prokaryotes have DNA. They have this nucleoid region. They have ribosomes. They have a cytoplasm. They have cell membranes. They also can have cell walls, which not all eukaryotes would have. Some things specific kind of to prokaryotes would be a pilus. You don't really find these in eukaryotes. That pilus can be used for a number of things. One of the things it can be used for is to exchange DNA between organisms. So notice here they're pointing at this plasmid. That's not just a circle, right? So that's not circling those ribosomes. That purple thing is the plasmid. Plasmids are just circular pieces of DNA. And it's typically in prokaryotes, those circular pieces of DNA is where you would find things that make that bacterium kind of more resilient. So let's say this bacterium was resistant to an antibiotic. That antibiotic resistance gene would probably be on that plasmid, right? And so that plasmid could be exchanged with another bacterium through that pilus, right? You also tend to find or can find these structures called fimbriae on prokaryotic organisms. That just allows them to stick together or to stick on things. It helps them form biofilms. You don't typically find fimbriae on eukaryotes. This structure, if you find a structure that looks like this on a eukaryote, it's probably cilia. And cilia help those organisms move in eukaryotes. You don't find cilia on bacteria or prokaryotic organisms. You can find flagella on bacteria. You also can find flagella on eukaryotes. They're structurally different and they move in a little different way, but they're similar. Right, so that's some of the major differences. The morphology of prokaryotes can range, and based on its shape, we give it a name. Right, so just a second ago I said Streptococcus, and so coccus for round. Bacillus or bacilli bacteria are typically rod-shaped. The Vibrio are curved. You can have them that are kind of intermediate, right? So Coccobacillus, which are kind of oval-shaped. You can have Spirillum or spirochetes. Right? And so those are all names given to bacteria based on their shape. Right? And so here we can go even further. So if they're round, they're coccus or cocci. But if they stick together in these chains, then we call them Streptococcus. And so all that tells you is, so Streptococcus tells you what it looks like. If they look more like these kind of clusters, those are the Staphylococcus. The Streptobacillus, right? So chains of rod-shaped bacteria. Right? And so that's where the names for them come from. They respond to changes in tonicity the same way that essentially eukaryotes would. And so you probably have been exposed to the idea of tonicity in, you probably had 168. So you've had some prerequisite for this course. But essentially that just points to the concentrations of solutions, right? And so, for instance, here they're showing you an isotonic solution. So isotonic means the concentration of two solutions is the same. So if I put a cell in an isotonic solution, water's going to move in and out the same, and so nothing's going to really happen to the cell. If I put a cell into a hypertonic solution, so everything out here would be the hypertonic solution, water's going to flow outside of the cell, and the cell's going to shrink. If I put a cell into a hypotonic solution, water's going to move into the cell, and it's going to move into the cell until one or two things happens. One, the cell ruptures because it just can't withstand the pressure, or two, the cell pushes against the cell wall to the point that more water can't go in. And so that's what's going to happen. This is also the reason why, so this hypotonic environment is why you wouldn't want to inject pure water into your bloodstream. If you inject pure water into your bloodstream, your red blood cells would swell to the point of bursting. Bad day. That's a real bad day for you, right? And so this is just showing you kind of what that would look like if a cell wall was involved, right? Nucleoid I talked about, some inclusions, you can read over those. Another kind of thing that bacteria can do that eukaryotes typically don't do would be this endospore. So endospores, if a bacterium is in a really harsh environment, say it dries out or whatever, they run out of nutrients, they can form these structures called endospores that can stick around for terribly long periods of time and then kind of come back, right? So an endospore could stick around for months or years until the right environment comes back, and then that cell would come back. One of the problems with spores is that when they start to grow, they tend to release some toxins too. Botulinum toxin, for instance, or Botox. That can come from those spores developing into vegetative cells. So if you ever wondered where Botox gets its name from, that's it. And the way it works, so Botox or botulinum toxin paralyzes muscles, and that's why it can be used therapeutically, right? So maybe they inject it over your forehead or in the middle of your eyes, and it paralyzes those muscles so that the wrinkles go away. But it can be used therapeutically too. So, for instance, I know individuals that maybe they have a calf for whatever reason that's just really, really, really tense, and so they walk on their tiptoe, inject the Botox into the calf, and it relaxes it, so they can actually heel strike like they should. But any number of things, stroke patients, right? So maybe their hand is constricted, can inject Botox to relieve that a little bit, so they're not clenching their fist so much, right? But those are endospores. Endospores are really hard to get rid of. That's why you have to make sure that your protocols for autoclaving, maybe in the hospital or whatever, you have to test that stuff all the time to make sure that you're reaching the right temperatures and the right pressures, and we'll talk about that later in the semester, because endospores are the real problem, or a real problem. Actually, prions are the real problem. It's really hard to get rid of prions. Prions are proteins, and one in particular causes mad cow disease, and so those can be really tough to take care of. What else? Let's skip ahead to the eukaryotes. Eukaryotes look a little more complex, right? So they have, some have cell walls and cell membranes, but then they also have this just really complex kind of endomembrane system. So a lot of membrane-bound organelles, be it the nucleus or the rough and smooth ER, the Golgi Apparatus, Mitochondria, Chloroplasts, right? So just a little more complex system inside. They can have cilia, they have flagella, they have structures that allow them to attach on to things. They have this nucleolus, not to be confused with a nucleoid. So it's just a region of DNA that's kind of concentrated inside the nucleus. But yeah, a little more complex. They can have things like lysosomes and peroxisomes inside. The other difference is with bacteria, when they divide through a process called binary fission, where eukaryotic cells will divide through a process called mitosis or meiosis, depending on if it's a somatic cell or a sex cell, right? And so really, the easiest way for me to remember this, and again, I've taught this for a number of years, so this might be why, what mitosis really means. So most of the time if I ask students, what is mitosis? They'll say cellular division. Technically not true, right? Mitosis is division of the nucleus, right? Well, since bacteria and archaea don't have a nucleus, they can't go through mitosis, right? Since the definition of mitosis is nuclear division. So the prokaryotes, when they divide, they go through a process called binary fission. I would encourage you to look at this kind of summary of cells and look at the differences and similarities between them. Maybe focus mostly on prokaryotes versus eukaryotes. We can talk about the bacteria and the archaea a little later too. But certainly look at, I might would ask you a question, for instance, like compare prokaryotes to eukaryotes. How are they similar? How are they different? Right? And this is a good chart to kind of look at that. Right? Morphologies of eukaryotes is all over the map. Right? They can be circular. They can be kind of these squiggly looking things. They can be kind of oblong. It's just a pretty diverse group of organisms, right? This is talking about the cell cycle and certainly mitosis. One thing to be careful of here is that we talk a lot about mitosis in biology classes, and so I think because we talk about it so much, students think that cells are most of the time doing it, and they're not. So if you look at the whole cell cycle here, so cell growth, DNA synthesis, more cell growth, this tiny little sliver is the only thing that's mitosis. So it only spends a short amount of time in that to divide. The rest of the time it's just doing cell stuff, right? So if it's a liver cell, it's doing liver functions. And so I think we have to be careful on how much emphasis we put on mitosis so that you don't get the idea that it's doing that all the time, right? What else? Talk about some individual organelles. So ribosomes make proteins. That's the gist there. The endomembrane system is the rough ER, the smooth ER, and that nuclear membrane. It's called rough ER because it has ribosomes on it. It's called smooth ER because it doesn't have ribosomes on it. Right? So pretty simple. The Golgi apparatus I kind of think of as UPS. So it kind of takes in things, modifies them a little bit, and then ships them out. To put it super simply, lysosomes are there to kind of break down things. So anything that's in your cell that needs to be broken down and gotten rid of, that's what the lysosome is going to do. So one way to remember this is lys, the root of that term means to break, right? So lysol, right? The reason it works is it helps break cells down. Peroxisomes help detoxify things in your body. So any tissues that you would expect to be responsible for detoxification, like in your liver, you would expect to see a lot of peroxisomes in those cells. Cytoskeletal elements, I don't really worry too much about differentiating these, although if you're in 169 and you're talking about muscle contraction, then really you're talking about cytoskeletal elements there with actin and myosin fibers. Right? And actually there it's showing you that, right? So the contraction of those. What else? I don't care about the difference between these. Mitochondria I mentioned a little bit before, right? So mitochondria is the organelle that creates ATP through the breakdown of glucose. It can use other things, but glucose is the most efficient. Right? You get a lot of ATP from glucose. Chloroplasts I mentioned, right? Take light energy and convert it into chemical energy. Helps plants make carbohydrates like sugars and store them as starches. Right? That's why you like potatoes, right? Because starch is a pretty high energy food and pretty easy to break down. Right? Some of those other stuff I've mentioned, cell walls. The extracellular matrix is just the stuff outside of the cell. Some cells have more of it than others. It just depends on where and what the cell is, kind of what its function is. So for instance, bone cells are going to have a huge amount of extracellular matrix, whereas muscle cells aren't going to have a huge amount. Some bacteria will have a lot of extracellular matrix to help create biofilms, where others aren't going to have quite so much. Right? And I think that gets us through kind of quickly the cell. So again, look over the summary, look over these review questions. I have you guys doing these open assignments. One of the things you could do if you're struggling trying to come up with an idea of what you should do for open assignments, just work through these questions, answer these questions, and turn that in to me. That's a good way to kind of study and then get some credit for it. Right? So if you have any questions, please let me know, and I hope these videos, I say short, this one's 30 minutes, I hope they're helping, and I'll continue making them.